The broadcast is now starting. All attendees are in listen-only mode.
Good morning, good afternoon, or good evening, wherever you may be joining us from today. You've logged on to the Nelson Labs webinar, Bioburden Alert and Action Levels, Low-Hanging Fruit for an FDA Citation. We will be joined by Bryce Telford, Expert Technical Consultant from Nelson Labs. Now, just a few housekeeping items before we get started. If you miss one of our webinars, or if you'd like to refer back to this one or any of them, you can always find them on the Nelson Labs website on the on-demand webinars page listed under the Education tab. You can also receive notifications for upcoming live and on-demand webinars by liking the Nelson Labs Facebook page or following us on Twitter or LinkedIn. Nelson Labs is a leading global provider of laboratory testing and expert advisory services for medtech and pharmaceutical companies. The company performs over 800 rigorous microbiological and analytical laboratory tests across the medical device, pharmaceutical, and protective barrier industries. The experts at Nelson know that every test matters and requires solutions to complex problems to improve patient outcomes and minimize client risk. So let's get started. A little introduction about Bryce. Bryce Telford is a recognized authority and industry leader on radiation sterilization, bioburden, microbiology, and tissue processing. He has over 13 years of experience working in laboratory functions and research, test design, media formulation, medical device, and tissue industries. Bryce works with clients in all areas of the world to speak, train, and consult on failure investigations, bioburden issues, establishing bioburden alert and action levels, product family grouping, product adoption, radiation sterilization, and tissue validations. So without any further time, I'll turn the presentation over to Bryce.
Thank you, Michael. So my name is Bryce Telford. So today we'll be discussing alert and action levels. And with that, what needs an alert and action level? When we check ISO 11737-1, it states acceptable levels for bioburden. So for the purposes of this discussion, we have bioburden that needs an alert and action level. Also, within the annex, it gives general guidelines for setting environmental or bioburden levels. And then in ISO 11137-2, it again discusses a review of environmental and manufacturing controls. So for today's discussion, we'll be focusing on bioburden and environmental alert and action levels. So definitions of what an alert and action level is. So we have the USP definition for what an alert level is, as well as an action level. So this slide's kind of here informatively. And also there are some additional references on defining what an alert and action level is. So from PDA, pharmacopoeias, and things of that nature. So these slides are for reference, then if you need to look back at the slide deck. For the purposes of our discussion and what we work with at Nelson Laboratories, we define them as follows. So an alert value is a value from monitoring, providing early warning of deviation from specified conditions. Another way we say that is that it might have drifted from normal operating conditions. So the product might have drifted from production norms, or the clean room, if it's environmental monitoring, may have drifted from normal operating conditions. And then the other side of that, we have our action level. This is a value from monitoring that necessitates immediate intervention. Another way of saying that is that it has drifted from normal operating conditions. So there has been a change, something has happened that is causing it to no longer be in normal operating conditions. So to also go with that, we have limit and levels. So some clarification there of how that's kind of used and defined within the industry is that a limit implies product has been impacted by the excursion that is above the value, whereas a level does not imply that the product has been impacted. It's a level that we are setting, but it doesn't necessarily mean that product has been impacted. As this kind of goes with the definitions, really what we're kind of looking at when we set an alert and action level is that we're looking for something where it might have drifted from the normal or it has drifted, but we want it to alert us and we want to take action prior to that affecting the product. So prior to us exceeding some limit. So kind of to visually think of that, we think of a level. So we have, for example, on the left, we have the motorcycle, and it's certainly exceeded the level of riders that it is specified to have, but it's still operational. It's still able to move around. They can probably get to the park and do what they need to, but it certainly has exceeded the level of riders it's supposed to have. Whereas a limit is a very hard definition. So we have a bridge, it has a specified weight limit, and clearly that limit has been exceeded. So we have exceeded the limit and we have a definite effect. So with environmental monitoring, when we're setting alert and action levels there, we have questions that tend to come up when we are doing alert and action levels for environmental monitoring. So if we exceed an alert level, should we cease manufacturing? Should we discard product? Well, the reality is that there's no direct correlation between environmental monitoring and those alert and action levels and actual product bioburden. So then that poses the question, why do environmental monitoring? Why set those alert and action levels? And as with our definition, what we want is to monitor the environment to make sure it's in its standard operating condition, its defined operating condition, and that if it exceeds that, then we're taking action prior to it having an effect on the product, prior to impact to sterilization, for example. So although there is not a direct correlation between environmental monitoring and product bioburden, we need to monitor the environment and we need to have those levels so that we can monitor it and take action if necessary prior to there being an issue with the end product. So alert and action levels, a lot of what we're going to discuss is going to kind of revolve around a few different sections with regards to bioburden and the radiation standards, particularly since we're mostly going to focus on those bioburden levels with also the importance for environmental as well. So when we're setting the levels, the guidance from the standard is to base the levels on historical data. So make sure that you have data behind it, and then use means or standard deviations to set the data. There's no industry-defined way, as we'll kind of discuss in a bit, but using averages, using means, using standard deviations in order to assess the overall production is recommended. And then with all of that, you need to perform continued trend analysis and data evaluation. Having alert and action levels don't really do a lot of good if you're not reviewing them regularly and ensuring that the product is within those defined levels. And then on top of all that, be mindful of what we refer to as spikes. And even if the average is within the levels or that you've defined, be aware of bioburden spikes. We'll talk about spikes in a little bit, but they tend to be an indication of an infrequent occurrence of a negative effect. So when we're setting our alert and action levels, we're needing to set it on historical data. So there's some considerations for gathering that data. And since it's my webinar, I get to have a little bit of a soapbox for a moment. And part of that is pooled test articles. There is certainly a time and a place for pooled bioburden testing. There are times when it is necessary in order to do that testing. But be mindful of pooling test articles when you're setting your alert and action levels and when you're monitoring your bioburden. There are some downsides to doing pooling. So there's no data regarding the distribution or the variation of your bioburden across individual units. and you're unable to detect as such individual spikes in bioburden. So be mindful of pooling your test articles and pooled bioburden testing. It does have a purpose when it comes to testing, but just be mindful of it. And if you really need to pool, or if you're going to be better off doing individual testing, what are your requirements for your testing for your particular product and the data that you're going to use? Also, when gathering your data, be considerate so that you have at least 4 sets of data that should be used. And with that, we recommend 10 samples per quarter, for example, for bioburden testing as part of a radiation dose audit. But initially, you probably want to have that higher level of testing even for other modalities, such as ethylene oxide, for example, so that you have a good statistical base in order to build your alert and action levels on. And with that, we set the alert and action levels. Those are a numerical value that we're going to compare to, but we also need to be mindful of what the nature of our bioburden is. So that's where microbial characterization comes in. As you're doing this data gathering process, it's important to start building your catalog of what the bioburden on your product is, not just numbers, but also types. And then with the product bioburden, we recommend employing a validated recovery efficiency. So generally our recommendation is that each product should have its own recovery efficiency. And that way you are comparing bioburden estimates for how the overall production is affected, so that there's also less variation there. And when comparing your bioburden estimates, so that you can see, is it an individual sample that is departing from the normal, or is overall production based on the bioburden estimate beginning to exceed that norm? So have that recovery efficiency, and we recommend having the recovery efficiency again for each product. Now, there can be some reasons and some justifications for why you might use the same recovery efficiency across multiple products, but be mindful of that and make sure that you have a reasonable rationale as to why you might have a single recovery efficiency, and just be mindful that individual recovery efficiencies is kind of more the recommendation and the industry normal. So when we're setting our bioburden alert and action levels, there's no requirement or established method for setting these levels. Common approaches are standard deviation, or 2 times the bioburden estimate, 10 times the bioburden estimate, looking at a log difference. There's no right or wrong way as is defined by the standards for doing so. So what you're doing is you're balancing between demonstrating the adequacy of control with doing too many or too alerts too often. So what we would refer to as the boy who cried wolf syndrome. We don't want to set our alert and action levels such that we are seeing too many alert and action triggers. After time, that's going to become very apparent that, number one, they're not set properly, and number two, that you're doing a lot of additional work, investigational work, things like that, that really is just being predicated on a improperly set level. So you want to balance having that adequate control, and what's adequate is going to be product and process dependent, and going to require some consideration and some definition and justification behind that adequacy. So what we discuss usually here is what we refer to as a common sense approach for evaluating the data. And we'll look at a few different ways of doing that, but a standard deviation isn't going to work for every modality that you're setting an alert and action level for. If, for example, it is a radiation sterilized, we might want to set a very tight alert and action level, and we might do a single standard deviation or 2 standard deviations. Whereas for something like ethylene oxide, that's where we might look at something more like 10 times the bioburden estimate or 2 times the bioburden estimate, different things of that nature. So taking a look at the data, what it's being used for, what your sterilization method is, or if it's environmental monitoring, how controlled you need the clean room, for example, and then taking a common sense approach for actually setting that alert and action level and which method is going to work for you. And there's many situations where we have used a variety of methods. So we might set the alert level based on a standard deviation and an action level based on a log. So it's also possible that you're going to be mixing and matching some. So evaluate the data and see what really fits for your product, what makes sense for your product, so that you're not triggering too often having that boy who cried wolf syndrome. So when setting the levels, standard deviation is pretty common, but we want to make sure that it makes sense. And a lot of times when we talk about it in statistics, standard deviations in microbiology, the microbiology itself isn't following a normal distribution. However, we found that the standard deviation is still a useful means of determining the dispersion of the data and making sure that we're accounting for that dispersion in setting our alert and action levels, so that we're accounting for what we know is regular background variation within our bioburden and our production. There are also statistical options for doing statistical conversions, such as switching from a normal distribution, doing a log transformation, things like that. But we found that even without any transformations, standard distribution does work rather well. And with regards to a Poisson distribution, generally the more data that we had, and if we had sufficient data, it wasn't found that when we analyzed large amounts of data that we saw that Poisson distribution. So that's again where it comes in that you have to have enough data and gather that data over time so that we're setting a reasonable alert and action level. So some general concepts when we're setting the levels and to be mindful of when we're also gathering the data is don't base product release or manufacturing on bioburden values alone. So for radiation sterilization, the dose audit is the primary driving force of acceptance or rejection of the verification, and therefore sterilization dose, and that's predicated primarily on your test of sterility. The bioburden is expected to see some variation. Why do we do the bioburden testing for a dose audit? Because we need to demonstrate that we have control over production and that that bioburden is within the range that we've established. So this is also a good time to bring up a level versus a limit. So if we have VD max 25, for example, that has a limit built into the table of 1,000 colony forming units. So we want to set our alert and action levels below that limit so that we're triggering before there's an issue with production. Now, if we're looking at our alert and action levels and that's not a reasonable possibility, then that might be an indication that we want to assess lowering the bioburden, looking into it, investigating additional actions, things like that. Also, again, environmental counts don't translate to bioburden counts, so we don't want to have an environmental excursion necessarily dictate alone the release of product. We want to look at the entirety of the equation and is there potential that the environmental impact is affecting the end product. But again, there's generally not a direct one-to-one correlation between environmental counts and product bioburden, so it's just one factor in that decision process. We also don't want to set the levels too soon. We want to make sure that we're gathering sufficient data and that we are able to account for things such as seasonal variation. And when we're looking at our alert and action levels, we don't want to establish the same level for all products that are manufactured. Everything that you make might qualify, for example, for say VD max 25, but that doesn't mean that you should set your alert and action levels for all of your production based on one kind of arbitrary alert and action level that's within that VD-Max 25. We want it to be based on that product and the variation we're seeing with it in its bioburden, so that we know we have control over that product. And then again, we want to check for the presence of potentially resistant microorganisms and start building that catalog of radiation resistance, for example, or if it's ethylene oxide, are we seeing a lot of spore-forming organisms? If it's other forms of sterilization, then we want to be mindful of what microorganisms we're seeing and what that might actually mean for the product and for sterilization. So that's something to gather as well and start building that catalog of the nature of your bioburden. So when we're setting alert and action levels, we tend to have a few items that we need to address. One of the first is too numerous to count. So when we have a too numerous to count result, we shouldn't use that data for setting our alert and action levels. We shouldn't give it a nominal value, so such as greater than 300 CFU, because we may be really underestimating the bioburden. What we should do is investigate the too numerous to count. Is it a true result? Was there a contamination event? Was there an issue with the test articles? It's sometimes possible to use reject items. Was that reject too much of a worst case and not representative? And then once that investigation completes into the too numerous to count, take corrective action that's appropriate and show that that corrective action is appropriate by doing additional testing. And since we're not able to use that data point, we need to do that additional testing also to get data that we can use for setting the alert and action level. So this is kind of one of those instances when we talk about environmental trending and monitoring for products. Generally, we talk about no, a too numerous to count result is a result, and that is the case. We do want to investigate it. We do want to take action. But from setting an alert and action level standpoint, and therefore a statistical standpoint, that's a data point we don't want to use, and we want to replace it with additional testing that has had that corrective action taken. So then we also have spreading organisms. So it may not be an accurate count. So without additional information, the count, again, should be discarded while gathering your historical data. Now, I say without additional information. There are situations where you are just going to see spreading organisms. Sometimes it's the nature of the organism itself that it's going to be spreading. However, usually it indicates that there's a problem with the test method. So we recommend considering changes or improvements to that test method. A couple of examples just for reference would be an auger overlay. So when we have a filter plate, we will overlay that filter with tempered molten auger in order to essentially trap the organisms. Another might be to use a higher factor. It's going to lower your limit of detection. It's not going to be as good of a limit of detection, but it may be able that we are able to get things in the countable range and minimize that spreading. Another would be to potentially pour plate, where we're again trapping it with an auger. But as mentioned, sometimes there's simply no improvement. You have spreading organisms, and that's where we mentioned the additional information. If you have those spreading organisms, we want to set the alert and action levels off of the most appropriate, most accurate data possible. If we're always going to see those spreading organisms, then that is when we would include that data. When we exclude the data, though, again, we want to perform additional testing so that we're collecting sufficient data to build our alert and action levels. And then, kind of lastly with some of this is spikes in bioburden. So what is a spike? So there used to not be a lot of guidance on this. With the updated bioburden standard, there is some additional guidance on what a spike is. So in the definitions, we have individual bioburden value that is significantly greater than bioburden values in a set. Well, that just poses the question though, what is significantly greater? When is it significantly greater? And that really is going to depend on the product and also it's significant is going to depend on how the product's sterilized or how that bioburden data is being used. There's currently no harmonized approach, but a common definition is an individual value which is greater than or equal to 2 times the mean. That's one option. What we recommend is making a definition for what you consider a spike based on your product, based on it, how it's being sterilized. If it's environmental monitoring, based on your clean room and what is being produced in the clean room, how critical that environment is to the production, and defining what is considered a spike. Normal, kind of just general one within the industry, is that 2 times the mean. So then when you do see a spike, we want to have that trigger some action. So investigate, identify, and evaluate. So investigate why that spike occurred. Is there a corrective action? Identify what that is. So on the example in the photograph here, we have a number of colonies, but we have predominantly one colony type. So identifying that colony, finding out what it is going to help with that investigation, and also may lead to some additional information that is beneficial, such as your sterilization modality. If it's a spore-forming organism and you're seeing a spike like this, that could be something that is going to affect the product. If it is a radiation-resistant organism, so you can kind of see that there's a pink and a red colony, those might be potential concerns for radiation sterilization. So identify so that you can use that information to also guide decisions and guide actions that the spike is going to trigger. And then evaluate. So if we have radiation resistant organisms and we have a spike of them, is that something that's going to affect sterilization? Is that something that is of concern? Evaluate the product and evaluate what that spike means for the product. And of course, consider your sterilization modality, whether it's EO, steam, radiation, so forth, or if it's something such as aseptic processing, things of that nature. So on the left, we have a data set, and sometimes a spike is very obvious. We look at the data and we can look and see, yes, that is clearly a spike. So we have that spike, it's easy to see, it's easily defined. But on the right, things get a little bit more complicated because we have variation in bioburden from, say, 20 to 154 colony forming units. We have quite a bit of variation there of what that is. So where is the spike? What is it that we're looking for? And this is why it's also important to define what you consider a spike. So if we use the 2 times the mean, we have a mean of 67.3. If we double that, then we can look at it and we can evaluate. Sample 2 is fairly high, but sample 10 is what would be considered a spike at that 2 times level. So we want to define what we consider a spike and take a look at it accordingly and do an evaluation of the data when we look for spikes. Spikes are separate from alert and action levels, but in many ways, if we see a spike, it's going to kind of do the same thing. If we see a spike, that could be a trigger to take additional action, do additional investigation work, things of that nature. So we have some different types of alert and action levels that we look at and we discuss. And one of them is the initial or the temporary alert and action level. Now, this is generally used when establishing levels for new product while you're in the data gathering process. Now, there's no industry requirement that temporary levels be established, but it is beneficial and suggested based on circumstances. So one example would be radiation sterilization. If you have just completed your 3-batch validation, you have 3 batches of bioburden data, you have some indication of what your bioburden is and how your dose was set. Now what does that mean for your very first dose audit while you're collecting that data in your second and your third dose audit? So that's where an initial or a temporary alert and action level comes in. So earlier I mentioned not setting your alert and action levels too soon. And this is kind of one of those exceptions because we are going to set it, it's going to be fairly broad, but we want some data, we want some indication of what the expected manufacturing normal for the product is, so that when that first dose audit comes along, we know and have at least an idea of what to expect and what is acceptable as far as variation in our bioburden. So use the available data that you have, and then based on the bioburden estimate or the overall average is how we're going to do it. We want, when we set the alert and action level, we want to see if manufacturer, we're looking at the product across basically a series. So for example, 10 samples to see, is it just one sample and a spike, or is it all samples that are seeing this increase in bioburden and therefore something that is part of the overall process. And then with the beginning, you want to consider some more frequent testing at the beginning to start gathering that data up front. So some examples of that might be to do basically like an initial batch by batch based bioburden testing on a number of samples in order to begin gathering that data. And then over the course of time, maybe a quarter, reduce that to being something like monthly bioburden testing. And then over the course of the year, you've gathered a month's worth of data for the entire year. And now we can scale it back to our regular testing routine, since we've gathered that initial upfront data and then we've scaled it back so that we can see how it relates. Now, when you set an initial or a temporary level, you want to as part of that, document a plan for setting your long-term levels and the frequency of evaluation. Far too often, an initial or a temporary level will be set, and then that just becomes the permanent alert and action level, whether it's because you forget, whether it's because somebody leaves the company and it's not, the next person isn't aware it was temporary. So as part of setting your temporary alert and action levels, if you choose to do so, there needs to be a long-term plan for setting your long-term levels. And also with that, you want to also indicate the frequency of evaluation and the frequency of your testing. So if you're going to have that increased testing plan to begin with, you want to have it defined how it's going to taper off and what that's going to lead to, so that you don't have excessive testing on down the road. The last thing you want is to, say, 3 years down the road, have a temporary alert and action level that's become default permanent, and also have that increased level of initial testing be your standard level of testing. So make sure that there's a documented, so that it can get to the next person in line, basically, plan for setting your long-term levels. So with those long-term levels, it's used once you've collected and set once you've collected sufficient data. Now, one thing that we're very mindful of is we don't use the term permanent levels. So we use long-term as kind of our definition, as our way of saying it. We don't call them permanent levels because the levels don't have to be set in stone forever. They should be assessed periodically to maintain the appropriateness of your alert and action level. As you continue to gather data, you can continue to refine your alert and action levels. There's also going to be changes to production, so your long-term levels can be part of that as evaluating through your change control. And they can also be very beneficial kind of going the other direction with your change control. So let's say you make a change to the product. You change raw material suppliers, for example. Having those alert and action levels is going to let you know what your normal baseline bioburden is, and therefore, does this change affect that bioburden level? In other words, if I have a new raw material supplier, is maybe a little bit of an increase in bioburden, is that something concerning from that supplier, or is that within my alert and action levels and something that I have defined as acceptable? Is it something I'm going to see? Now, if we change suppliers and we immediately have, say, an alert level, that's not a great sign, and that might be an indication that some changes need to occur, maybe to the supplier, to the process, or even just changing suppliers, something like that. Or even potentially needing to trigger a dose audit and looking for radiation, for example, and making sure that your sterilization dose is still acceptable. So it can work both ways. So you might need to reassess your alert and action level when you make a change, but it can also help you to evaluate that change as well. You want to make sure when you're setting your levels and when you're gathering the data that it's representative of the entire lot. So common practice we see is beginning, middle, end. So a number of samples are taken from each time point in the lot. So with that, some extra care and consideration is needed with simulated products or products that are made specifically for a validation or for testing, so that when you're setting those levels, it is representative of normal production. So have some extra consideration there. And then we want that data to represent common variation. So earlier we talked about making sure, and here we talk about having at least 4 data sets. However, along with those 4 data sets, we recommend having commonly a year's worth of data as well. And if product's not produced on a regular basis, maybe it's produced every 6 months, getting those 4 data sets might take 2 years. Whereas if you do it on a quarterly basis, you might be able to get it all entirely in 1 year. So be mindful of both of those, but we want to have a year's worth of data so that we have that seasonal variation. The environment and the nature of your bioburden is going to change over the course of the season, summer, winter, autumn, spring, as well as potentially there's going to be other changes. So water system. Here, for example, at Nelson Laboratories, we're located in Salt Lake City at this facility, and we have a combination of surface water and groundwater. Some of the year we're getting surface water, some of the year it's coming from wells, and there's going to be differences in that water. It's going to meet it's whatever the city guideline is, but you're going to see variation, and therefore you're going to want to see that accounted for in your bioburden. You're also going to see changes in maintenance as well as training. So you might have somebody that's brand new at the start when the validation is done. Is that the next person that comes along going to be properly trained, properly know how to do it? Or are people also going to pick up bad habits, things like that? So we want to have that year's worth of training accounted for as well. You might have different suppliers depending on the time of year. And with all of that, we want to make sure we're accounting for all that seasonal variation, all those various changes that are going to happen over the year, small changes here and there. So that's why we also recommend that year's worth of data, is so that we can see what that variation is. And then again, we want to employ a validated recovery efficiency when possible and applicable. Now, I say applicable because there's certainly products and bioburden tests, for example, where we don't do an extraction and therefore there's no recovery efficiency. So that's what we talk about applicable. So let's talk about the common sense approaches to take. So some considerations when setting the alert and action level is really, first of all, is this an environmental alert and action level, or is this a product bioburden alert and action level? If it's environmental, we might have constraints such as clean room requirements. If it's bioburden based, we might have additional constraints such as the limit of a VD max table. But we also have to be considerate that with environmental levels, as was mentioned throughout this, that there's not a direct correlation to product bioburden. So we want to have those levels there for monitoring. We want to make sure that we have control over our environment, but it's not necessarily going to trigger an issue when it comes to sterilization. So it might have different triggers of what an action level means or what an alert level means and what action you're taking at each point. So be mindful of that. The next is to be mindful of your sterilization method. So overkill methods versus bioburden-based methods. If it is ethylene oxide, if it's or something of that nature, you're going to be killing off 1 million of the most resistant organisms, and then you're essentially doubling that. So you're seeing a massive reduction, and it's based on the most resistant organism. So you have a lot of additional flexibility when it comes to your bioburden. So set your alert and action levels accordingly. We want to make sure that we have control over production. That's why we set the alert and action levels. But we want to make sure we have quite a bit more flexibility too, to account for that difference in sterilization. Now, within bioburden-based, there's also some additional considerations. We want to set it tighter because any variation in bioburden is going to have a potential impact with our sterilization. Now, as far as flexibility goes, generally kind of the breakdown is you have a little more flexibility for VD Max than you do for method 1, and you have more flexibility for method 1 than you do for method 2. Now, there are, of course, within VD Max some additional considerations because there are the different VD Max levels. So if you're using a lower VD Max level, such as VD Max 15, that's not going to have the same flexibility that, say, a VD Max 25 has. And there's also the method 1 lower doses table as well. So you're going to want to be considerate of kind of all of that. You're looking at the entire picture on how much control you need to demonstrate versus how much flexibility you want to give yourself so that you're not having too many triggers. You also want to be considerate of your product and the nature and source of raw materials. If you have a tissue or tissue-based product, you're probably going to see some additional variation in your bioburden, but you're also probably going to have a fairly low dose. So you're going to need to balance both those considerations. The same goes with if you have natural materials such as cotton in your product. You're going to see variation in your bioburden. Does that mean that you need to have some an even tighter alert and action level to ensure that your sterilization isn't affected? There's also going to be consideration if there's kind of a final process. So if you have tissue, for example, and it's going through an antibiotic treatment, you're going to be wiping out most, if not all, of the bioburden. The same if you're going through a chemical treatment, such as a glutaraldehyde treatment, you're going to have different processes that are going to lead to essentially additional control over the bioburden. So that might give you some differences in flexibility. So you're going to consider all of this. You're going to look at the big picture and take that common sense approach to balance things out when you're setting your alert and action level. Generally, it's pretty common to err on the side of caution when you're doing your first kind of setting of your alert and action levels. And you might get going through the process and you might find out that you've set it too conservatively, or that you're having too many triggers. And again, that's where you are going to continually assess the alert and action level for its appropriateness. It might be found that you set it very conservatively to begin with, and that works just fine. It might be too conservative, so you need to reassess the data and see what works for you and your product. So with this, we have the basically kind of an overview of bioburden data from this table. So we have our overall average, which generally is our aerobic and fungal averages added together. We have a recovery efficiency and we have a bioburden estimate. So with that, we also threw in some additional data. So we have our standard deviations and our bioburden estimate and what those look like. And when we're taking a look at our common sense approach, a lot of times what we're going to do is take a look at this data and see what makes sense for your product, but also what makes sense microbiologically. So a common approach, as is mentioned, is to use standard deviations. And it might be that you do bioburden estimate plus 2 times the standard deviation, bioburden estimate plus 3 times the standard deviation. And rounding to an extent is also fairly common. So what we might do is set our alert level at, say, 200 colony forming units and our action level at 250 colony forming units. Well, within the average standard deviation, we're going to see those 50. And also microbiologically speaking, is there really a difference between 200 colony forming units and 250? And that's where we have to consider our sterilization modality. So for example, if I'm doing VD max 25, I have some additional flexibility, and 200, 250 really isn't a lot of change in the bioburden from a microbiology standpoint. So that might be a case where we would look at something more like, is it 2 times the bioburden? Because when we talk about dose setting, it's often if it's 2 times the overall average, then we use a different, we use a single batch, for example, for bioburden for setting the dose. So we might look at that. We might look at things logarithmically. So sterilization is logarithmic based. So if I'm seeing a average bioburden estimate of 81.4, 1 log higher, so 810, that's going to be a log increase, and that is definitely where we would expect to see an alert. No, I apologize, an action, because now we've gone higher. We've gone a log higher. We now know that a log is quite significant because that is, when we're talking sterilization, it's logarithmic based. So when we see the bioburden go a log higher, chances are pretty good that we can see something when it comes to the sterilization. So we certainly would want to set our action there, for example, or less. So that might be an upper limit. But let's say we're working with ethylene oxide and we're talking about an overkill method. Well, if we see a log increase in bioburden, that's certainly a change, but we're talking about a overkill method where we're looking at essentially 12 log reduction in overall bioburden. So when we have those 12 logs, is a 1 log increase in our bioburden that's already several logs below, and we're also considering this is our standard bioburden versus the most resistant organism. Logarithmic approach doesn't necessarily make sense. So that's where we start looking at things like the bioburden estimate times 10. So we want to look at the data. It's good to actually have kind of a table set like this where you can look at the data as a whole and take a look and see what makes sense based off of my data for my product to demonstrate control without triggering too many alert and action levels. So consideration, again, number of types of routine bioburden. If we're seeing a trend or a change in the nature or of the product or the environment, that's something that we should be considerate of. We want to look at that overall trend. So we want to identify the recovered microorganisms, see what we're looking at. We want to evaluate those microorganisms for resistance. So for EO, that might be looking more so at the spore-forming organisms. For radiation, we're looking at the known radiation-resistant organisms. We want to look at production personnel and potential impact. So these are things that we want to look at when there's an alert level trigger. Are our personnel properly trained? Do we have new personnel that don't have a good understanding of what the process is or why they need to do things a certain way? We want to look at, has there been any manufacturing changes? And we may want to evaluate our sampling and our testing procedures. Are we using reject samples? Might not want to do that. Do we need to increase our sampling for a period of time to show that we have returned to a normal level? Also with that, an evaluation of laboratory controls and monitors. So it does happen. Laboratories make mistakes by all means. So we want to have an investigation into the laboratory and into the testing. Are these true results? Do we need to consider additional testing? Maybe we had a initial sampling of just 3 samples and we exceeded our alert level. Do we need to do additional testing? Maybe we need to send in 7 additional samples to get us up to 10. Do we want to test additional batches? What do we need to look at? So there may be consideration for doing additional testing at an alert level to kind of come up with your scope of potential impact. And then if nothing's really found, generally a thorough cleaning of production area is pretty common, changing out gowns, doing a thorough cleaning, changing out various equipment, cleaning it, things like that. And then do we need to change or modify our sampling plan? So within radiation sterilization, for example, there's dose audit frequency reductions where you can reduce, instead of doing quarterly, to doing it every 6 months and then to doing it just once a year. If we have this alert level excursion at our bioburden testing though, do we need to change our sampling plan? Do we need to do our dose audits a little more frequently so that we're demonstrating that we still have sterilization control over the product? And then whenever there's an alert level event, kind of the first place we always look is water. Is there any water, or can we find the source of contamination? So all things to kind of consider when you have your alert level that it might trigger actions to take at that alert level. Now, when we have an action level excursion, we're going to want to do all of the stuff that happens at the alert level, but we're also going to have a requirement generally for a determination of potential impact on sterilization, and there's going to be a root cause analysis and investigation that has a corrective action for returning production or returning the environment to the proper bioburden levels so that it's no longer exceeding that level. So you do all the environmental work with the alert level, plus you have the determination of impact to sterilization and that root cause analysis with corrective action. So some do's and don'ts with that is certainly don't base all of your release of products based solely on these levels. These are levels that we're setting. It is based on data so that it has some meaning behind these alert and action levels, but we don't want to base product release solely on these levels. There are other considerations. So with ethylene oxide, as an example, it's an overkill method. We have our product challenge devices. We have our biological indicators and things of that are really our indication for acceptable product release, whereas our alert and action levels are us making sure that production is under the appropriate level of control. So could it cause us to look at and evaluate whether we should release product? Absolutely, but it shouldn't be solely based on just the bioburden. There should be other things to look at, other things to investigate. Do we need to do a dose audit? Do we need to do some additional testing? Do we need to do bioburden on additional lots? But it shouldn't be solely based on product bioburden and meeting those alert and action levels. And certainly products shouldn't be discarded based solely on the bioburden. We've seen situations where we have the alert level and products held, and we have the action level and products discarded. That's a pretty extreme situation when you're basing it solely on bioburden. So it really depends on what that bioburden data is being used for and what your sterilization method is. So you don't want to just start discarding product based solely on product bioburden. But what you do want to do is consider the impact on the sterilization process. And that brings us to kind of one of the big considerations for overkill is, as was mentioned, there's a 12 log reduction of the most resistant organism. So when we're setting our alert and action levels, we're usually looking at it to give ourselves quite a bit of flexibility accordingly. So some standard approaches might be to do a bioburden estimate plus 3 times the standard deviation. So not just 1 or 2 times the standard deviation, but 3 times the standard deviation. Other common approaches are 5 times and 10 times the bioburden estimate. And again, this is all about giving yourself the proper flexibility, because there's really no effect to sterilization because of that 12 log reduction of the most resistant organism, but we're defining that there's been a change to the product, a change to production that is affecting bioburden, and we want to take action in order to return it to its controlled state. So kind of in summary with all that, our alert and action levels, they are required. It's part of bioburden. It's part of environmental monitoring. So it is required to have those alert and action levels. It's pretty common that an auditor will come in and one of the questions is, well, how do you know your bioburden is under control? How do you know this bioburden is acceptable? And that's where the alert and action levels really come in, is that's where you're basically giving your answer of, well, our bioburden is properly under control because it's within our alert and action levels. If they need to see how the alert and action levels were set, you make sure that you have the documented rationale behind kind of why it's there and why it's appropriate. And again, use a common sense approach, gather the necessary data over time. Spikes are separate from alert and action levels, as are TNTCs, but they're definitely things to be investigated and to watch for when you're doing your trend analysis. And it may be necessary to make changes to your test method in order to eliminate, for example, spreading organisms. Then with all of this, you want to document a course of action and perform periodic reviews. The last thing you want is to have an alert level excursion or an action level excursion and then go, okay, my bioburden has exceeded the alert level, now what do I do? If you don't have anything defined, if you don't have anything set up, then there's really no plan in place. And in a lot of situations, it's going to look like potentially that you're just kind of making things up in order to fit what the outcome that you need, or you're going to be forced to take some very conservative approaches in order to avoid the appearance that you're kind of data selecting or anything like that. So document your course of action ahead of time. It makes it a lot easier to rationalize why you did what you did instead of it looking like you did what you did for some other alternative reason. And then again, don't release product based on your alert and action levels alone. Have the other considerations there. What's my sterilization method? What am I using this data for? Is this environmental data? What's the nature of my bioburden? Am I seeing radiation resistant organisms along with this increase? Am I seeing staph epi along with this increase? Look at the other factors. Don't base it just on the number and being within the alert and action level alone. So with kind of all of that, alert and action levels tend to also lead into dose audit frequency reductions. So even though the dose audit may be reduced on its frequency, there's still the expectation of bioburden testing being done at least quarterly. And that's a very important place where these alert and action levels come in, because then since you're not doing that quarterly dose audit, that bioburden becomes that much more important, that much more critical, and demonstrating that control therefore becomes that much more important and critical. So that's where these alert and action levels really come in. With a family grouping or a product adoption for sterilization, one of the requirements, for example, when we're doing a family group or a product adoption for radiation sterilization is the bioburden comparable? And one of the best ways that we can assess that is if we have products that are within an alert and action level, it makes it a lot easier to adopt that product in, for example, or to begin to build that family group, because we have defined what is kind of an acceptable alert and action level based on that. And then it's also a very good tool for any failure investigations. We can see, have we had a trend in the bioburden? Have we exceeded alert and action levels? Have we taken action? And really the entire purpose of our alert and action levels is that we want to take action prior to it leading to that failure. We want to take corrective action. So for some additional information and resources on our various offerings, we have Nelson Laboratories for advisory services, our sister companies Sterigenics and Nordion, that can also be reached out to. If you have any questions, feel free to reach out to their sales associate using the contact information below. And we offer a wide variety of services across the three companies. So if you have any questions, feel free to reach out to your account rep, reach out to the emails listed, and we can certainly get you pointed in the right direction and see if that's an offering or something that we can set up. Michael, with that, I turn the rest of the time over to you for any potential questions.
Great, thank you, Bryce, and thank you for all that great information you've shared with us today. We do have we have received some questions. I know we're about out of time, so we'll get to one or two of your questions, and then Bryce will be able to get to you offline if we don't get to your question. So let's just get into this here. The first one, Bryce, is how do we establish alert and action levels when typical counts are low but are not necessary for the sterilization dose applied to the product? The data is not normally distributed. Parenthesis, many bioburden samples have no growth, close parenthesis, and the product is gamma sterilized, validated with VD Max 25 method.
So that's very product specific, and it is something that we fairly commonly see, though. You're doing VD Max 25, you have, say, less than 3 for your aerobic, less than 3 for your fungal, and you're seeing that less than, meaning we're seeing just the limited detection. We're not actually seeing organisms pretty commonly. And when we're setting those alert and action levels, it's a good time to evaluate how the product is being tested. So do we need to change the bioburden method to make it something that is more sensitive so that we can see what our true bioburden is? But also, that's not necessarily the case. We're already just showing a very low level of bioburden. So that's where we might take an approach where, rather than using standard deviation, because there really isn't any standard deviation, we're seeing less thans, we might take a different approach, such as doing something like 2 times or 5 times your bioburden estimate. And that's where, for radiation sterilization, the log comes in handy. Is since it is since sterilization is logarithmic based, that's where if we're seeing less than 3, that's going to end up less than 6. For just example purposes, let's say the recovery efficiency is 100%. So our bioburden estimate we're routinely seeing is less than 6. So we might want to set our action level at, say, a log higher, where we might be doing 60. It really also kind of depends on some of the other variables too, such as what are we seeing over time, as well as the nature of kind of the product. Is this a tissue product, something like that? So that's something that we could certainly help offer some additional guidance with if we set up a project. But generally, it's one of those cases where instead of using a standard deviation, we might look at one of the other approaches.
Great. Let's just take
Next question.
one more question here, Bryce. Let's go with, which reaction against the product do you recommend if bioburden fails?
So bioburden fails is kind of a general term. It's really going to depend on, for example, your sterilization method or what you're using the data for, as well as what you set your alert and action level at. So if you're doing radiation sterilization and you exceed the limits of the table, there's some additional guidance offered in 11137-1. It usually triggers additional testing, such as an immediate dose audit or things like that. Now, if we're talking about an alert level where we've exceeded that, then that's kind of in the previous slides, where it's do additional things, such as do your bioburden investigation, have the product bioburden identified. With that too, when you're doing your identification, you might want to add some additional specification for what IDs you're looking for. If you're looking for radiation sterilized product, it might be something where you're looking at, for example, pink, red, orange colony forming units, so that you're seeing if there's anything radiation resistant that you need to be particularly concerned about, things of that nature. So it really kind of depends on what you're considering a bioburden failure. And we handle failures. That tends to be a consulting project that we do, so that's something we can offer additional guidance on. where we can customize, kind of tailor it to your product and situation.
Great. Well, thank you, Bryce, and thank you for all this information, and thank you to the attendee for attending this webinar. We would hope that you found this information and this session valuable in your experience. And if you have any questions, like Bryce said, please reach out to Nelson Labs or any of our sister companies with that information that you have. Again, we thank you, and we hope you have a great rest of your day.